Wai-Yim Ching. I have been with the University of Missouri, Kansas City for more than 40 years. So I'm very pleased to present this talk, which is started only about a half year ago. And my topic is the large scale of initial simulation for biomolecular systems, in the case of spike protein for the SARS-CoV-2 virus. So I first will briefly introduce the motivation. Everybody knows that the COVID-19 pandemic poses a severe threat to the human health and unprecedented social and economic disruption. So this COVID-19 offered actually a once-in-a-lifetime opportunity to address some of the most difficult scientific problems. And so this S protein or spike S glycoprotein is the most important one because it makes the initial contact with the ACE2 receptor in the human body, and is the key to understand the virus anatomy. So here's the picture about the spike protein, and this picture actually shows that this blue triangle is this spike protein. So all the others are the structures of the SARS-CoV-2, And we want to using computational methods to challenge this very interesting and very important scientific problem. So I first will make a very brief outline. I will say a few things about the introduction, and will introduce what the spike protein in the SARS-CoV-2, and briefly mention about the computational methods, and then this will be followed by the results. And the results are divided into 5 different types. First, our strategy of studying this S protein is using a divide and conquest strategy. And this is just an outline. And then we will first describe the structural optimization of the structure of the protein itself, and the main result on the electronic structure and also the partial charge distribution will be discussed, followed by intra and intermolecular bonding in those domains. And last, we will briefly mention about the sequence conservation, which is very important in the biological system. And then I would briefly summarize the conclusion of what we have so far accomplished in the last 6 months. And but more importantly, I would say something about the work currently in progress, which consists of the non-local three-dimensional amino acid interaction, and also the computational of the mutation effect and the drug design. So the introduction is that I want to say it a lot straightforwardly. Biomedical science is an interdisciplinary field which consists of all the science areas, including of the basic biology, chemistry, physics, mathematics, material science, and computer science, and pharmacology, and medicine, you name it. I myself was trained as a condensed matter theorist. My research area now sort of expanded into computational biophysics. That's why I'm giving a talk here today. We usually use the density functional theory-based quantum mechanical methods, and we have to use the most advanced supercomputers for those large-scale modeling. So the goal is to provide the key information on the structure and the properties of these complex wireless systems. And admittedly, this is a very narrowly focused area of COVID-19 research. You hear from the news almost daily on many different aspects of combat COVID-19. And so the main goal is to collaboration with other scientists, especially the experimentalists, is the key to the success, because I myself is the computational theorist. I will discuss recent progress and also on the non-local three-dimensional interactions between amino acids and its implications on the sequence conservation. And this research also provide a fundamental understanding of the virus structure and a pathway to the design of effective therapeutic drug design. So here is the spike protein of this SARS-CoV-2. It actually consists of 3 chains, A, B, and C. So this is the view of the 3 chains varying from the top. And the chain A, which is in the upward conformation, is the most critical one. And this is the structure is first published in the Science by Dr. Rupp and co-workers. So each chain actually consists of subunits, which is the receptor bonding S1 and the membrane bonding S2. I would explain a little bit more carefully in the next slide. S1 have 4 domains, which is NTD, RBD, SD1, and SD2. And then S2 consists of 3 domains, okay, which is FP, or the fusion peptide, and H1, HR1CH, and CD, which is a connector domain. So this is very wordy, sometimes it's not easy to remember, but this is how they are named. So this is again the same slide to show these areas, the chain A I already mentioned, and then there are 7 structural domains that in this chain AS protein. So this is the computational part for each of them. So NTD, SD1, RBD, SD1 in S1, FP, HR1CH, CD in the S2. So it's rather complicated, but again, this is the main result or main talk of about these 7 structural domains that I'm going to present. Yeah. And so another similar to be figure to make them easier to understand, amino acid in the 7 structural domain in the S protein. So this part is S1, this part is S2, and these are the 4 domains, and these are the 3 domains in S2. And so these are some of the details about how many atoms, how many amino acids in each of those domains. And those black vertical things are the ones that the amino acids are missing their coordinates, so those are simply omitted in the calculation. So this is posing some kind of difficulty in the real calculation. So now I move to the computational methods, So we use 2 density functional theory-based methods for the simulation. The first one is a very popular package called WASP. Many of the people in the area, in this computational area, probably knows it. And the plane wave-based method, specific potential choosing, and it's very efficient for structural relaxation and the mechanical properties calculation. The second DFT method is the orthogonalized linear combination atomic orbital method, and this uses atomic basis expansion, and it's one of using the LDA, and it's very efficient for complex large systems calculations. We calculate the what we call the total bound order density, and so this method is described in the book I wrote, and I will not go into the details. It's developed in my laboratory for last 30 years or so. And the most important thing about this method is the actual quantum mechanical calculation used on the molecular scheme is called the bond order between every atomic pair. So you have two atoms, alpha and beta, and you solve the quantum mechanically. You have all the alpha level integrals and wave functions, and you can calculate the bond order values. And if you add all the bond order values together, divided by the specific volume, you get a total bond order density, which is a very important parameter. Simultaneously, another very important quantity to be calculated is effective charge Q star, which gives you the charge on each atom, and again, using the molecular scheme. And these days, we expanded this bond order calculation concept into the concept of amino acid bond pairs, AABP. Okay, this is essentially you can expand your results calculated from the bond order values to the interaction between pairs of amino acids, because in biology, the basic unit that people are interested in is the amino acid or residues. So on the results part, I've the first just presented the one of the domain, which I will not going to present, which is one of them is the first paper we published in this area, which is available in the PCCP, the journal, and of the Royal Society of Chemistry, and about several months ago. And so it is, again, describe the structural refinement, electronic structure, partial charge, inter and intra-atomic bonding, and sequence conservation. So this is one specific domain. In this presentation, we cover all the 7 domains. So the results on the 7 structured domains S protein. So I've divided this into these 5 topics. The first one is the divide and conquest strategy. I simply just list the strategy itself. So we started the calculation for each of these 7 structured domains, just like in the RBD, in the previous slide. These results are then collectively presented for all 7 of them, analyze them, and link them in a meaningful way. And we could be able to obtain some conclusions reached, and critically discuss them and try to understand the entire S protein. So this is what we call a divide and conquer. You calculate 7 of them independently, and you try to combine them in a very meaningful way. So now I go to structural optimization. And again, the structural optimization is of the S protein is actually determined by the shallow electron microscopy method experimentally, I already mentioned. And with the PDB data, deposit those coordinates. And so the amino acid sequence are all deposited in this PDB data, and but some of the coordinates of certain amino acids are missing, and due to some technical difficulties. But those are the experimental results. Is not a concern with what we are doing. And also those PDB data, the position of the hydrogen atoms that terminate the dangling bonds are missing because people just assuming there are hydrogen atoms there. So such structures published experimentally in top journals like Science and so forth I'm sorry, I don't know why I'm go back to so this can give you very beautiful figures of the structures of, but it's not enough to do the adequate ab initio computations, because in the ab initio computations you have precise atomic positions for every atoms you are calculating. So our first step is to refine the atomic positions to its equilibrium structure. And we have first added the hydrogen atoms, with a Camilla program, fully relax the structure for each domains to higher accuracy. And we have to use the supercomputer from NERSA and of the Department of Energy, and using the WASP code. And the large number of each domains, require a huge amount of supercomputer time. For example, in the NTD domain, it has almost 3,500 atoms, and RBD, which one is the published paper, is 2,100 atoms. So we estimate that the positions of each atom has the precision of about 0.01 angstrom. So these structures are they used as an input to perform the DFT calculation using the OSCO method I mentioned earlier. So here's the results very quickly. So here are the results for these seven seven structural domains. Listed these seven structural domains already mentioned: A, B, C, D, E, F, G. And in the first look, there I have a very similar structure, but actually there are some sort of very subtle differences. And of course, I'm not going into the details. And those results would be available to anybody who are interested. And the most important part on the electronic structure of partial charge is the partial charge distribution. So these are, again, these seven structural domains: A, B, C, D, E, F, G, NTD, RBD, SD2, SD1, and FP, HR1CH, and CD. And this is the entire domain on the solvent accessible surface of the amino acid with large PC. So some of the amino acids, those are marked with their sequence number, and some of them have a very large positive charge. Some of them have a very large negative charge, partial charge, as shown in this colored bar. So this information is extremely important and very difficult to get. And we were able to sort of decipher the details of the partial charge distribution. So this is one direction of the view, but this is a three-dimensional structure. So the next slide shows the 180-degree of flip around to see how they look like. So after flip by 180 degree, you can see this. There are different part of the large partial charge, positive or negative, and those even those partial charge on the surface or solvent accessible surface is very important in the actual interaction. So here's the intra and interatomic bonding for this seven structure domain. So this again looks very similar, but they have a very subtle differences. And you can also notice every type of interatomic bonding have been indicated. Most of them are strong covalent bond, and then there are hydrogen bondings, and there are some different pairs of types of atoms. So I have to move a little bit quick because my time is limited, and so this is a very important study because it concentrates on the hydrogen bonding. And most people doing ab initio calculation, hydrogen bonding is extremely important in the biological systems. but most people do not know the details about the strength or the distribution of the hydrogen bonding. So you can see that there are very different type of hydrogen bondings, and here we showed this hydrogen bonding from different domains, and NTD, SD1, and so forth. And the most important thing are the hydrogen bonding, so which have the bond order values bigger than 0.1. Those are very strong hydrogen bondings, and this could play a very critical role. And then there are for the hydrogen bondings more higher than the 2.0 Armstrong apart, they are much weaker. But the problem is that they are ubiquitous. There are huge amount of this. So when you add them together, those hydrogen bondings also play a very important role. So the take-home message is that our method and calculation can decipher into all the details about interatomic bonding, include especially the hydrogen bonding. And here are some examples. I'm not going into the details. So I said the details can be omitted. Essentially, I give two examples of specific interatomic bonding in one of the domain, which called RBD. So, this is one amino acid, this is another amino acid, this is a third amino acid, and how they interact with each other, how big is their the bond order values, which one has the hydrogen bonding, and so forth. So this is just two examples. The fact is that we have all the details of every one of them and every of the amino acids. So this A and B are just some figure captions. I don't expect you to read in this very fast presentation. So I said details can be omitted. So now the last part I'm talking about is sequence conservation. This is a very busy figure on a very novel topic. Only people in the biology are very interested in the sequence conservation. The sequence conservation essentially tells you the arrangement of the amino acids on the different or very similar type of the materials. For example, here there are 5 laws. The first one is the COVID-2, the SARS, the one, the protein we are talking about. Then here's the beta-1, beta-2, SARS-CoV, the older one, and the beta-3, those are from the bats, okay, animals, And you can see they have the amino acid sequence almost the same, but there are differences. So the details is very complicated. So the important thing is there is a blue box here, which is from the paper by Anderson in the Nature Medicine. Those identified as to be highly conservative sequence. Conservation sequence is very important because it is related to the mutation, it relates to the evolution, and it relates to the fundamental part of the virus mutation. So I will not go into much of the details. And again, so I try to explain that the figure of the previous figure, but it is just too much details involved. So I said the details can be omitted in the presentation. If anybody who are interested, you can find this in the PCCP paper I mentioned earlier, or you can send me an email. I would try to answer your questions. So now I come down to the summary and the conclusions of what we have achieved. Basically, we optimize the structures of 7 structure domains in the S protein using WASP. This is more accurate than purely from an experimental measurement from a cryo-electron microscopy measurements. We performed the detailed ab initio DFT calculation on these 7 domains using the fully relaxed structure. And then we reviewed the atomic scale bonding information about the hydrogen bonding and for in the chain A of this protein. And the partial charge on every individual atoms and every amino acids are obtained in the structure unit, and the potential correlation with sequence conservation in the RBD has been reviewed. I have a typo error here. So we are in the process of developing a new technique called three-dimensional amino acid interaction, which is one level deeper than the interatomic interaction between every pair of atoms. We want to connect them into the amino acid, and which has to be a three-dimensional picture. So the method we developed can be applied to the study of the mutational effect of amino acid, and the design of the drugs currently are now in progress. So now I want to shift the gear to say something about what we are doing right now. Okay. So this is the last part in my talk. It probably will take me another 5 minutes. So this is the work in progress. So the work in progress is the three-dimensional non-local amino acid interaction, because these are not the local one between the nearest neighbors. And this is a new concept introduced called the AABP, which in the method section in the last equation, I describe how they can be calculated. And we are going to study the interfacial interaction between two biological units. One SE2 is in the human body, one is RBM, is a part of the virus. So we wanted to do our initial investigation of the effect of a special mutation, which is very controversial or very popular, is D614G. 614 is the amino acid sequence in a protein data bank. D means one of the name of the amino acid. After mutation, it becomes G, because all the amino acids, you can have a single letter to describe them. So D and G, okay. At the interface of the S2 of the S protein. And so computational design of the drug and the vaccine is also the work currently in progress. And of course, we are computational scientists. We sort of main reason of my talk here is to try to seek cross collaborations with some experimental groups. Hopefully, we can give some useful input. So here's a very busy figure, and I want to look at the details, the description omitted in this presentation. And this is, again, some kind of a strategy to put a three-dimensional amino acid interaction onto a two-dimensional, onto the two-dimensional page, which is very messy. Nobody can actually understand. Even in the submitted paper, they really have a difficulty to understand them fully. So I would, if you have, I simply show how complex they are. And these are 144 amino acids, each with a circle. These are the primary sequence. They are linked to each other. But then there are non-local interactions linked by those lines, so all in a three dimension. So I apologize for the details, but again, I will try my best if you have questions, and I can answer to you. And this is the same thing, about the details to be omitted, because this is describe the actual AABP data on this 144 amino acids in the RBD. And you can see those yellow ones are the nearest neighbor or the primary sequence. And then the gray ones are the off diagonal, which some of them can be very important and have some very unique features. Of course, they are much weaker than the nearest neighbor, and because everybody just using the nearest neighbor sequence, including those sequence conservation, all the nearest neighbor primary sequence. So I would not go into the details in this presentation about these results. So the working progress, another one I just mentioned about this mutation is this mutation of within one of the unit, and this is somewhere between RBD and SE. There are some other mutations, and these are the work we would be currently planned to do. And then there's another thing is the interface model. So this is the interface between SE2, which is a cell in the human body, and then the RBD. And you can see very clearly there are this interface between them, which is the very important part, because whatever drug you design is try to put them at the interface. So I have this very nice figure to see about this interface, about their rotations. So here are the 4 drug models. Currently, we are trying to attempt the computational research. So D1, D2, D3, D4, and most of them probably heard about them. So D3, D4 has to do with chloroquine, hydrochloroquine is actually the recent news is they are less effective. So we are focusing on the drug 1, drug 2. So those are the molecular formula of this drug, and put them at the interface. And these are very large models for us to compute. And it will take a tremendous amount of supercomputer time. So this is my last slide, and so some acknowledgement. These are my team members. Some of them are here today. This is Dr. Adhikari and my PhD student, Baha and Salo, and also Ms. Namek is a medical student, So we have other collaborators. Some of them are experimentalists. And we get the financial support from National Science Foundation, which is a special award called the RAPID, and that means you have to do fast, So we have the tremendous amount of computer support from supercomputer, the NERSC of the US Department of Energy, and also from the computing RSCC from University of Missouri. And here is my email address, and anybody who want to contact me, send me an email, and I will send you whatever information you want, or what kind of suggestion or criticisms, please send to me. Thank you very much for your attention.